Hello, everyone, and good afternoon. It's my pleasure to welcome you on behalf of the EFFoST Working Group on Sustainable Food Systems to today's webinar, where we want to dive a little deeper into the aspects of food safety together with sustainability and what are new developments in that field. I am Felix. I'm an assistant professor for food process engineering, and I'm the chair of the Sustainability Working Group. And together with Arthur Robin, who is also here today, we organized this session and we identified some very skilled and very interesting speakers. So we are very happy to welcome you all to this interesting webinar today. And before we get started, just a few words. So as I said, we want to talk about the interface of food safety and sustainability, starting with a talk of Annette Winkler from Cargill on re-emerging new pathogens that are caused or that comes through the fact that we have the climate change happening now. Then we have Annette Recek-Jambrak from Zagreb, who will give some implications on mitigation strategies, how to inactivate, how to control pathogens in a sustainable way to ensure that we have safe food in the end. Then we have Uli van Aar from Agroscope Switzerland, where we want to look into the new challenges by the raw materials, and especially when we look into the plant-based alternatives, which are, of course, more sustainable, but can come with some different challenges compared to the original product, let's say. And last but not least, a very important topic that is always present when we talk about both hygiene, food safety, but also sustainability. And this is the whole fact of cleaning and disinfection and also hygienic design and how this affects on the sustainability of the food processing and how we can also maybe optimize this a little bit to improve the sustainability even more. Yeah, maybe just a few introductory words on the working group. What are we? Who are we? What are we doing that way? Basically, the working group got together several years ago, around 5 years ago, as we saw that we wanted to start some more initiatives for the food science and technology community. Especially since we all had some issues with the fact that sustainability is often just used as a buzzword or for greenwashing purposes. And we try to put more scientific evidence into this discussion. And we also have the strong aim to communicate and to provide opportunities for our research bubble, let's say, to meet and to discuss, and also to provide content and information for people outside of our research bubble. This is the idea. And so we are a group of experts and scientists working in different fields related to food science and food technology. You can see the new working group configuration now. We just added some new members and we want to welcome all of them, of course, today with this webinar. And just a short look who we have in the group. So me and Arthur, you know already. Then we also have Henry Jaeger from our Boku University in Vienna. Annette is also part of our panel today. So we will hear more from her later on. Then we have Kelly, who is from the food industry. We have Sergey and Beatrice, who are from the German Institute of Food Technologies, working a lot on sustainability quantification. And then our three newest members are Clara, Byron, and Alexa. And they are all junior researchers, upcoming researchers, very talented people, also researching the fields of sustainability and sustainable food systems. And once again, warm welcome to you all. Okay, that being said, now it's time for our first talk. But before we get into that, just some housekeeping. You may have noticed that this is not a traditional Zoom call, but this is a webinar version of Zoom, so it works a bit differently than you may know it from your other meetings. So if you want to ask questions, this is possible through the Q&A feature that you should see on your screen. Please put your questions in there in the written form. We will then select the questions and ask them to the speakers. Also, once the speakers are done with their talk, they also have the possibility to answer some questions in written form if this is applicable. Yeah, that being said, it's time now to introduce our first speaker, and I would like to warmly welcome Annette Winkler from Cargill, who will talk about the re-emergence of pathogens. And I will quickly share the screen. Annette, you can already unmute yourself if you want. And you should now also see the screen, I hope.
Yeah.
Good.
At least I can see it, Felix. So
perfect.
thank you.
Then, yes, thank you very much. The floor is yours.
Thanks for moving the slides and helping me here. And thank you for inviting me to talk. Now it's about 20 minutes to talk about a subject that can easily fill a day, I guess. So what I will try to provide you with is some food for thought, how climate change affects pathogens, spoilage organisms, and in general, maybe microorganisms, which are interacting with us and living with us in the world. So with that, maybe to the next slide, please.
No, just a moment now.
Yep. So the WHO had published their food safety strategy, and most of you will be very familiar with that. And it lists drivers of change in food safety. And the red arrow points you directly to the topic of the day, environmental changes that relate to accelerated or accelerating climate change and others. So that's setting the overall framework when you think about climate change and food safety recognized by WHO. And with that, we can go to the next one. So then the thought is, what does climate change entail? And there are a few publications on that. I like the one with the mountain on the left side, where you can see it starts with a climate variability. So it really starts with you have a changing climate you did not expect that links to extreme weather events. And you see them listed. You've heard the news, most likely, most of you following that up. Also natural calamities, again, that comes to extreme weathers like tsunamis, monsoons, cyclones, floods, and droughts. Now, you see, the question is, yeah, how do microbes survive? How do they grow? By adaptation. Yeah, and there are already some examples that have been published how microbes adapt when they cannot escape those conditions. For instance, microbes in soil. They adapt their enzymes to deal with higher temperatures, and they also adapted to other changes in terms, for instance, of heavy metal uptakes. So there are quick adaptations happening in the microbial world that might also have an effect at the end on our food supply. But it's all about adaptation. We'll need to keep an eye on. So next slide, please. Now, it's not only food that is affected by climate change or food microbes. It's microbes and anything in general. So also public health as such is very interested in climate changes and the impact on emerging infectious diseases. Here is a very nice graph. You can see what they pulled out. What are the environmental outcomes of climate change? That comes from the former slide. You already remember there have been heat waves, so increased temperatures, erratic rainfalls, or increased droughts. So it's either floods or no water at all. They also related to pollution and also to a decreased ozone layer with increased UV. Now, that in response comes with expanding the reservoir of some of the pathogens, let it be the animal reservoir or let it be simply the geographical reservoir. Because a changing temperature, for instance, also in the oceans or in the seas, means that some microorganisms who were not really able to replicate because maybe the temperature was too low, they now start replicating again in areas where they have not been on screen or in screening so far. And that can go back, interestingly, to the last cholera outbreak that happened in Hamburg in 1895. I don't know how many of you have read about it, but it was an interesting one where Robert Koch made some of his postulates. And that was caused by a very heated summer in Hamburg at that time. And the temperature of the Baltic Sea there were above 20 degrees Celsius, which usually is never the case. And that allowed the Vibrio there to multiply in a much higher rate than before. And in conjunction with very bad hygienic conditions that led to a cholera outbreak, a really disaster that time for Hamburg. Having said that, we know that those Vibrio cholera are still in Hamburg, they're still in the port, they're still in the water. However, they are kept down in their multiplication by temperatures. Now, imagine temperatures increase again and they are more stable at that temperature. What's going to happen? We need to have a better safeguard then to prevent another outbreak. So that is, as you can see, everything is linked when you think about climate change. It's not only food, it's our overall environment, and it's coming from everywhere. WHO also took some measures, as you see, to prevent or limit transmission event, but I'm not going to go into details. Whoever's interested, look at the paper, it's very good. With that, let's go to the next one with the limited time. We talked about outbreak weather events and coming back to food, focusing on food. Already in 2018, there was an interesting outbreak in Australia that was linked to cantaloupe, to the melon, Listeria monocytogenes. And when they looked into the reasons, you can see it with the red, let's say, in the red buckets. There has been adverse weather events. There was a heavy rainfall followed by a drought, so that the melons was exceptionally, let's say, dirty. And whatever the company had been doing that time to remove the dirt was not sufficient after those weather events. And therefore, finally, when people cut the melon, they introduced the listeria to the melon. And because of the high pH in the melon, it didn't die. And yeah, people got sick. So that was one of the, let's say, I would say first linked events to weather conditions or weather changes and a foodborne outbreak. But let's look at what follows and what else do we have. Next one, please. The recent outbreak. Again, it's melon, but this time it's salmonella. It's coming from Guatemala. And again, when you think about it, Guatemala weather events is hot and it's humid. So there is an investigation ongoing. So I cannot tell you the root cause, but you see two things. First of all, melons, and melons pop up quite often because of their outside. They have a little bit of rough surface where dirt and also microbes can hide. So when you're not cautious, or when you face some unusual events, then those melons are easily contaminated. In the next slide, I think comes more of a link to salmonella and climate change. That is an interesting study done in Australia, where they looked at patterns to see what mainly drives basically the salmonella risk in food linked to weather. And you see it on the graph here that the temperature is the main driver. An increase in temperature about 1 or 1.5 degrees is said to reach about a 10% increase in salmonella infections, if not basically treated accordingly. Humidity and rainfall are less associated with an increase in salmonella, but what is associated with salmonella is heavy rainfall in terms of flooding. So again, extreme weather events is something people need to be aware of, and you need to be vigilant if you are in production, if you're in government, if you're in public health under those events, not only immediately, but also what happens to the food produced during that time. Next time, please. This is just an overview. Salmonella new vehicles since, you see, 1970. It's fairly a busy slide, but you can see is a lot of materials or products that had not been in scrutiny a while ago. So clearly cocoa and chocolate starting 1970, but last big outbreak was in 22, if someone remembers that one from the Kinder Surprise. Then also herb spices, especially nuts or nut spread. The last one depicted here is 2025. That was pistachio cream. 22 was peanut butter in the US, quite big. 23 tahini or something related to sesame seeds. Now, honestly, that's not the last one. I should have, because tahini and sesame seeds are notoriously known for a linkage with salmonella. So usually every 6 months you see something in there. Flour and salmonella is also not a good association. But again, also flour, those are coming from the field. Everything of those, they come from fields at the end of the day. So again here, weather conditions, animals living there, harvest conditions, storage conditions, they all play a role. If they change, and if you change the origin, then be vigilant. Look at what can happen. Then the other ones are like breaded chicken nuggets, dry pet food, have also been involved in such outbreaks. Breaded chicken nuggets is interesting because usually people consider that those are fully cooked, but they might be raw inside and need to be cooked. So that's also something not directly linked to climate change, I would say, but maybe to consumer behavior. But can be more linked to climate change, clearly, as you see all the vegetables. Cantaloupe I mentioned before, but especially onions, tomatoes, cucumbers. That really depends on where and how they are grown. And I come to that later. Interesting notes are, it's not only the climate change as such, but it's also the way people grow the crops, people irrigate their crops, obviously with water, and where does this water coming from? I marked one in red, which is the moringa leaf powder, and I think that's going on the next slide in more detail, which is an outbreak that's more or less ongoing. It was closed and then it was reopened, and there were several Salmonella cereus involved in it and are involved in it. Those are capsules from, it's basically a powder from a plant, and that's been transported through the whole world. Now, with that, this is not a novel food, but it's supposed to be a healthy food, and people started to consume this kind of food only, I would say, in the last maybe 5 years or so. So also this kind of change in consumption has an effect on our, let's say, outbreak situation and on public health. Now, as if this is directly related to climate change, I don't think so, but it might be related to the fact that this is more and more consumed by a lot more people, and also the way it's being processed and transported is quite a long supply chain from where the plants are grown, then are they dried, then are they made into powder, and then they reach the consumer. So again, it's to keep in mind the supply chains that are transporting our food and how they are affected further, or how they are controlled is also very important. Then to the next one. Pathogenic E. coli, similar to Salmonella. You see listed several vehicles. Again, you see lettuce, onions, cucumbers. I want to focus on those. Those are again vegetables, and there was an interesting outbreak in 22 in the UK that was a contaminated lettuce, and here they really looked at the risk coming from climate change, and that's more outlined in the next slide, please. That is from the publication, which was a very interesting study they've done, and I think it was also one of the first studies where they used AI because of the immense amount of data they looked at. Yeah. So in this region in the UK, they have normally sheep. And then they looked at the timing and found that just before they had heavy rain with surface flooding, and the contamination happened exactly most likely with the irrigation water. And the irrigation water is usually taken from surface water. So it was water that was runoff from the floods. And then even when you try to wash the lettuce, there is a slight reduction when you wash it, but it's not like a validated kill step or a validated pathogen reduction step. So there can be some microbes still on the lettuce when you purchase this kind of washed lettuce. And that obviously made the people sick and led to the infection. And again, it was one of the first studies that linked climate change in terms of flooding and irrigation water towards an outbreak of pathogenic E. coli. Next one, please. Now, I talked about bacterial pathogens, but here is also fungi. And again, it's not only related to food, also those hurricanes and air movements also lead to higher fungal infections of the pulmonary tract and of wounds and of the skin, which is outlined here. So just for you to keep in mind, climate change and effect on pathogens is not limited to food itself. But on the next slide, there's an effect on the food, not directly by fungi, but via the mycotoxins they form. This is a world map where you see the different, let's say, prevalence. It's not exclusive, but it's kind of prevalence between the different Fusarium and aflatoxins. And clearly, when you do have climatic stress, that leads to insect attacks, plant disease, and can again lead to a higher proportion of the crop being infected with those molds, and then in consequence lead to higher toxin formations. Next one. So what can we do then? Yeah, I mean, we need to find a new balance between everything. Of course, yeah, stopping climate change, okay, that is out of my expertise, so to speak. However, what we need to keep in mind that at the end of the day, the only constant in life is change. So we need to be aware. We cannot sit down and say, okay, what we do this year is valid next year. We need to challenge ourselves and be open-eyed to see what happens around us, what happens in the world, to be able to basically provide or continue to provide safe food to all the people. And I think the last slide is kind of summarizing what we could do, looking at safe practices and validated processes that is within food production. We also definitely need research. We need to look at processes. We need to look at new technologies. We should employ AI, use it as good as we can in the limits it can, but that can help us towards the surveillance of the environment. And there are already EU projects underway looking at how weather forecasts can work with farmers to look at their, let's say, at the potential of mycotoxin formation or protection of their crops. And also consumer education is another part of it. That all can be expressed in a dynamic risk management board, but I don't think I'm going to go into details here, but I'm going to finish. As promised, I thank you for your attention, some foods for thoughts, and think about change is the only constant. Thank you.
Yeah, thank you very much, Annette, for your interesting insights on this very relevant topic. Since I was sharing the screen, I unfortunately could not have an eye on any questions. Arthur, were there any questions coming up so far?
So far, no, but I invite all the participants to add the questions. Myself, I added some, so I can shoot at it until people add questions. Feel free to add them. I have a question for you. You wanted to say something, Felix?
Actually, I noticed one person was raising their hand in the from the participants. Can you please write in the chat what you want to share with us? Thank you. Sorry, Arthur. Please go ahead.
No problem. Yeah, I'll start with a question for you, Annette. You mentioned a bit about all the different causes and the fact that a lot of things are related. So weather, flood, air, change of practice, and that bring first things to my mind is, can we actually prevent that, or what you say, we are a bit doomed to only be on the reactive side of like, should we invest a lot of time on preventing while it seems it can arrive from anywhere? Or from your opinion, it's better to build a system when we can react fast to newcomers?
I think when you see something completely new, you need to react. But then when you've seen it once, and we would hopefully be able to make connections towards what's the true root cause, then it's always good to be proactive and look, can we either prevent the root cause, or can we at least give a forecast? And that's a lot what's happening now, also in the EU, different projects to look at when we use all the data we have, let it be on weather, let it be on water, let it be on certain pollutions, can we provide a forecast how this would affect certain, let's say, parts of the overall food chain? And that could be to animals, and that could be to crops. So I think the future for me is more trying to use the immense data we can gather to provide a forecast and prevent it from happening. Because when I can work with farmers to say, hey, you need to seed now, or you need to harvest now, and that prevents mycotoxin formation because it's at the right timing, yeah, then that's the best we can do. Prevention is always cheaper than cure. How good we are successful, I really I cannot tell. But we try.
Exactly. Thanks for your answer. Indeed, we'll do our best, but potentially with the AI and big data, we have at least a fighting chance to be able to forecast.
Yeah.
Maybe also a short question from my side, if that's okay. Annette, I was wondering a bit. You were showing the different impact factors on the pathogenic growth and how it's influenced, particularly the temperature, the humidity, and so on. Now, I was wondering, was there also some reportings of other microbial growth factors that are changing? For example, different solubility of oxygen, promoting, for example, more anaerobic growth, or something in that direction. Is this also known?
Not to my knowledge, but with the wealth of literature that's available, Felix, I cannot say it's not existing. Definitely in the few ones I've seen and I looked at, where that mainly was the focus on salmonella, that was not mentioned, yeah. But you're absolutely right. Certainly that will also have an effect. And nutrient availability, when you think about the runoff, yeah, or any sewage that goes into water, into soil, certainly. And their antibiotic resistance might be a bigger one.
Yeah. Okay, thank you very much. Do we still have some urgent questions?
We do have we do have a question.
Great.
I will put it there. Maybe we'll need. I do not fully understand, but I will give a try to read it, and then let me know, Annette, if you don't understand it. So it's Elam Karimi that writes, "Thank you very much for this very interesting talk. My question is, what do you think is the main driver of this bacterial outrage? Outrage? Is it primarily the chemical compounds involved, or it is related to the nutrients being provided?
Mm-hmm.
Also, in your opinion, what would be the best first step of research direction to further investigate this mechanism?"
When you think about bacterial outreach, then is it the thought about the increased growth or spread of those bacteria? And I think that's more linked to also the capability of being spread. So water is an important medium to spread them. And then with the water, the temperature and what Felix mentioned before, also the nutrients, the runoff into the rivers. There are interesting papers, and there are already predictions being made using AI, and those link to specific water areas and the spread of Vibrios or of Salmonella in those areas linked to weather and nutritional composition of the water. So I know at least of 2 studies where they say, well, we do those predictions, and the predictions are about 85% correct. That's what they say. So there is and that was made for public health in the first place, because summer is coming and they were looking at where people go swimming, obviously, and there are outbreaks when people go swimming and have pathogens in the water. Some may remember I think it was the Olympia in Paris also, somewhere there was something like that. And so they tried to prevent that by doing some predictions where they can say, well, it's dangerous to go swimming or bathing in those waters. Again, it's predictability about 80%, linked to water, linked to specific points where they take and look for nutrients in the water. So it's a combination of all. And I think the future research should focus on more an integral view, not only looking at water, not only looking at temperature, but really AI gives the capability to combine all of those, overlay, and then see if the pattern makes a fit.
Okay, thank you.
Maybe one last short question from the Q&A section.
Yeah.
How does the climate change have effect in the production of agriculture products in Asian countries, especially in India? Do you have some facts about that, Annette?
I have not searched for that, I must admit. So if you want really to have facts, I will need to reach out and I will need to check for publications. Now, what is the case with India is the case with all the world. You have very varying weather conditions, and India might be lucky because sometimes they have like 2-3 harvests per year. So maybe one harvest is gone because of a weather event, but still that means a lot of hunger for the increasing population there. So I think those rapidly changing weather events, and in the past where they have very stable monsoon seasons, I've seen this in the past 2 or 3 years when I talk to my Indian colleagues, they are not that predictable anymore, and that causes problems also for the farmers.
Yeah, then once again, thank you very much, Annette. It was very interesting insights how everything is connected to a bigger picture. And now with the next talk, we want to look a little bit more how we can encounter these pathogens and these contaminations. And for this, I'm very happy to introduce Annette Recek-Jambrak from Zagreb. And Annette, if you would like to share your slides.
Thank you, Felix. Yes, I will share the slide. Okay, hope you can see the slides.
Yeah, perfect.
Okay, good afternoon to everyone. I hope you can see the slides and not the content above. So you are screen sharing and stop sharing. So hope this is not the case. Thank you very much for this invitation and this really nice webinar. Today I'm going to talk about non-thermal technologies for microbial control in food processing. So to look over the sustainable and innovative food preservation. So my name is Annette Recek-Jambrak and I'm coming from University of Zagreb, Faculty of Food Technology and Biotechnology in Zagreb in Croatia. Of course, as we heard, microorganisms cause spoilage and foodborne diseases. So the idea is definitely to destroy them or to make them not to have so good conditions to grow. So traditional preservation uses heat. We all know this, preservation, so pasteurization, sterilization, but heat can reduce nutrients and flavor. So non-thermal technologies can be alternative to provide safer alternatives. So the main action is to treat our matrix, our food at lower temperature, so in between 35-40 degrees. And with food preservation, so we without usage of high temperature, so we can maintain quality flavor and nutrients and also extend shelf life. The idea is to improve food safety. Of course, we have different strategies and also mechanisms. for our non-thermal processing techniques. So we have pulse electric field, we have cold plasma, UV irradiation, already known for years, for decades, for a pulse light, of course, ultrasound technologies, high pressure processing, ozone treatment. And of course, we have all the advantages and disadvantages of those non-thermal technologies. But of course, those technologies are eco-friendly alternatives to thermal methods. So of course, we are scientists, so we are often in the lab working in the technology readiness level between 1 and 4. And if you want to step out of the lab and if we want to scale up, so we are moving from technology readiness level from 5 to 9, going, of course, to the industry. And of course, we need to see what's happening with the non-thermal processing market. So we see that there is an increase in the market of non-thermal processing. And of course, it will continue to grow, especially with the two technologies like high pressure processing and pulse electric fields that are already introduced in the industry and very efficiently working in that area. So, of course, we can apply our non-thermal technologies in dairy industry, food and vegetable industry, meat industry, egg industry, fish industry. And of course, we can, what is also beneficial, we can use them as hurdle technologies. In example, we can combine advantages and use of advantages of each of those. So it's, in my opinion, the future. Major non-thermal technologies, as I already said, are HPP, PEF, plasma, UVC, ultrasound, ozone, and we have different mechanisms of their action. So main mechanism for HPP is high pressure, for PEF, electric pulses, for cold plasma, reactive plasma species, for UVC, DNA damage by light, for ultrasound, it's cavitation. Of course, we have key sustainability benefit in terms of lower energy consumption, reduced chemical disinfectant, lower energy use, and of course, in example, improved extraction efficiency. For HPP, we have different industrial application, and of course, it disrupts microbial membranes. Examples can be used for juices, guacamole, sauces, deli meats, and etc. So for a pulse electric field, of course, we all know that there is high voltage pulses applied to liquid. It creates pores in microbial cell membranes and causes leakage and cell death. So different liquid food that can be treated by pulse electric fields. For plasma, we have different ionization of gases that generates reactive species, and it damages DNA, proteins, and membranes. And of course, it can be used on low temperature and of course for surface treatment. But of course, it can be used for different matrices. But anyway, we need to be cautious because it needs to be applied for food, but we need to be cautious because this free radical formation. For UVC treatment, we have, of course, a very long time that UVC is used for microbial damages, and it prevents microbial replication. Of course, different applications, water, juices, and surfaces. Of course, ultrasound, high intensity sound waves. The main effect is cavitation, and this bubble collapse damages microbial cells. Also, it can be used in liquid like milk processing and extraction. So we have a variety of examples where HPP and PEF can be used for fresh juices, maintaining nutritional values and flavor, and it's commercially used worldwide. So also, cold plasma ozone reduces microbes and extends shelf life. Of course, we have different advantages and disadvantages, but we need to have this main effect on inactivation of microorganisms. What are the sustainability benefits of our non-thermal technologies? Are low energy consumption, reduced food waste, also minimal chemical preservatives, and supporting sustainable food systems. So we have also seen the impact on sustainable development goals, and also we need to see the larger impact in terms of environment, economic, and societal impact. So lower energy consumption, in example, for comparison with thermal pasteurization, where we have high temperatures. Here we have ambient or mild temperatures. Of course, there is significant reduction in energy demand and lower carbon emissions. Of course, in example for PEF, there is example of reduced energy use by 30 to 50% compared to thermal pasteurization. Here we have examples of industrial application of HPP, and of course, what is really important, longer shelf life, higher consumers acceptance, less product spoilage and waste. Also, we protect our nutrients and, of course, texture and sensorial benefits. Also, there is a lower need for chemical preservatives and benefits that we can use, in example, clean label foods, and a lower environmental impact for chemical production and disposal. Also, non-thermal technologies help maintain nutritional quality, and also reduction the need for nutrient fortification or processing losses. And also we have what is also important, water and resource efficiency. Also, what is also really important, less process and water and cooling, and this improves resource efficiency. So potential for continuous application, we have increased efficiency, reduced processing time, and reduced energy per unit of food. Comparison of different technologies, different mechanisms, different application, of course, in packaged or without packaging. What is really important to mention here, because we are using ambient temperature, so we are not using high temperature, we are not destroying directly, but indirectly with different, in example, oxidative species, and with mechanical forces and with pressures. Here we can see viable but not culturable state after application of non-thermal technologies. So they are alive and metabolically active, but cannot grow in standard culture media. So anyway, we can think that we have, okay, we have 5 log reduction, but in example, after 12, 24 hours, we see the increase in numbers of our microorganisms. So they are alive, but we cannot see them, and they may resuscitate, and this can create hidden food safety risks. So we need to be really cautious and use a different type of strategy to see, in example, to see them, and really to watch on these common bacteria that can enter, like Vibrio, Escherichia, Salmonella, Listeria, Campylobacter. So we need to be really cautious. So they are under stress because we are applying different stress effects, like oxidative stress, high pressures, and really they also have low nutrients, and they of course want to fight back to survive. So we want to inactivate microorganisms, but of course we can injure cells instead of killing them, and this can lead to this VBNC state. So we need to be really cautious. We have different application of, in example, non-thermal processing, like PEF. So cells may be killed, or sublaterally injured. This can lead to VBNC state. Also, cold plasma, we can see reactive species like reactive oxygen species, reactive nitrogen species. But of course, sometimes cells survive and enter in VBNC form. So we need to be really cautious. Possible results after UV and irradiation: dead cells, injured cells, VBNC bacteria. So what are the problems? False negative microbiological tests. Plate count shows no growth, but bacteria are still alive. So we need to not to underestimate these microbial risks. So we need to use different methods, like fluorescence staining, qPCR, flow cytometry, different analytical tools to detect what is happening with our microorganisms. So what is the future here? In my opinion, so we have, of course, advantages. We have limitations of our non-thermal technologies. Advantages are quality preservation, energy savings, but limitations, equipment costs, poor resistance, and of course, some methods are limited to liquids or surfaces. What else we need to think about when we discuss sustainability in broader terms, like environmental benefits? So we need to use the software tools to valorize if something is sustainable or not. And of course, life cycle assessment can be applied to determine if our non-thermal technologies are sustainable or not. Of course, regarding environmental benefits, we need to think about lower greenhouse gas emissions, lower water consumptions, so reduced energy, lower processing losses, reduced packaging waste. and of course, longer shelf life. So we have examples, what are the benefits of thermal processing, but compared to non-thermal processing, we have lower energy consumption, high nutrient retention for non-thermal processing, lower water usage, lower food waste, and also we can upcycle food waste, and also smaller environmental footprint. So LCA is really important for our non-thermal technologies in terms of electricity consumption, equipment manufacturing, packaging requirements, food shelf life, and waste generation. So by LCA, we are able to determine the total environmental footprint that is actually lower than conventional thermal processing. So of course, what can we measure? Carbon footprint, CO2 emissions, total energy consumption, total water use, nutrient pollution in water systems, emissions, and of course, use of non-renewable resources. Comparison with temperature, in example, non-thermal are near ambient temperature, lower energy consumption, and of course, a lower carbon footprint, and shelf life is very good. So to conclude, so non-thermal technologies can improve food safety, maintain nutritional and sensory quality, support sustainable food processing, and also we have different open area to innovate for the future food industry. So we can improve, and we need to improve resource efficiency by using renewable sources. Of course, we need to use more sun. Of course, we can reduce carbon footprint. We can reduce food waste. Also, we can reuse, in example, agro-food wastes and byproducts. So we need to upcycle them. And of course, we need to enable high quality, minimally processed food. So these technologies are aligned with global sustainable goals like UN Sustainable Development Goals. So we have 17 of those, and we can definitely tackle each and every one. So regarding here non-thermal processing, so we are targeting responsible, of course, consumption and production, climate action, but of course we are targeting other goals. Also, I would like to share the book that we have published about non-thermal processing in agri-food biosciences, where we discuss about non-thermal processing and, of course, sustainability of biomaterial production. Also, this is a book that was published in May this year, and it discusses AI interventions in agriculture, food, and health sector. So really the future is here, and we just need to use our technology wisely. And thank you very much for your attention. And also what I would like to mention, if it's not safe, it isn't food. So thank you very much.
Thank you very much, Annette. Very nice insights into the novel emerging technologies for food processing. Let's have a look if there are any questions in the Q&A section. Not yet. So this is again our invitation to you, to the audience. Please, you have the unique possibility now to ask an expert in the field about whatever comes up to your mind. So please use this opportunity to write your question in the question and answer section. In the meantime, maybe I can ask a short question, Annette. To my experience, the novel technologies, they are all suitable for certain niches, let's say. I think the conventional technologies, especially heat treatment, is unfortunately something that is very versatile, and that this versatility very difficult to replace by default, let's say. So it's quite challenging to find the right use cases for the individual novel technologies. And what do you think, what would be a good way to approach this, or how to, let's say from the industry perspective, if someone wants to come up with a new process or want to improve the sustainability, how can they achieve this, given the fact that also in the science, let's say, not everything is researched that well for the novel technologies also. What would be your suggestion for someone in that situation?
Yeah, thank you. Yeah, this is really important question. Of course, we know that thermal processing like pasteurization, sterilization is really the thing that is working, and we are sure that this is supplying to us really safe food, and that we are secure in that manner. So regarding non-thermal technologies, so they are here in the last 20 years. I have been working with those technologies from 2006, so for 20 years now. And there is the huge effort that is taking place with high pressure processing and pulse electric field. And really, for those technologies, they are proven to be safe, to be focused, to be really precise on the food preservation and to increasing in shelf life. And there are evidences, of course, regarding juices, regarding, in example, for HPP meat delis, a different variety of food. So in my opinion, as we have seen the market increase, of course, there is innovation in each and every sector. I think that, in example, each industry has its own niche where there are experts. And in my opinion, of course, each non-thermal technology is very precise and focused on different, on specific metrics. So, of course, we have advantages and disadvantages for each technologies, non-thermal technologies. And in example, ultrasound cannot be applied on some pulse electric field can. And of course, we need to really check first what can we do with our technology. Have we chosen wisely our technology? And what is the output? What kind of product and what kind of, in example, shelf life? What kind of product in terms of course, food safety is the most important thing. But of course, from the aspect of sustainability, what are we looking at? So lower energy consumption, in example, treatment with in-packaged material. So we need to see really what is the output. So in my opinion, we have a huge technology, we have experts, and of course, we know the advantages. I think that we need to be really precise. What are we going to do with our technology? And I think that the solution is in combination of non-thermal technologies and of course in hurdle technologies, of course in example for the future in combination of thermal and advanced thermal and non-thermal technologies. So to be sure that we have inactivated the microorganisms.
Thank you, Annette. In the meantime, we also have a question in the chat regarding the VBNCs escaping non-thermal processing treatments. Can dual non-thermal techniques or a combination of low thermal and non-thermal treatments eliminate the occurrence of VBNC?
Yeah, we have done the research of VBNC while using plasma technology on the different species like Salmonella, Escherichia, and we have found that, in example, after plasma treatment, after, in example, 24 hours, we have seen the increase in number of bacteria. So we have done the analytics, and we have been sure that those bacteria were under stress due to, in example, oxidative species. And so, of course, they fight it to survive. And in my opinion, the combination of non-thermal techniques and thermal techniques can be the solution to inactivate microorganisms. Because we have seen, for in example, like PEF plasma, that we can obtain VBNC state with for our bacteria, and for that we are not sure that we have inactivated microorganisms. And if you want to have some proof that your technology is working, so you need to show to FDA that you have viral reduction, so that you are able to complete the inactivation and that you are able to have stable inactivation, that you actually did the inactivation. If we see the occurrence of VBNC, so we are in that case not sure. So in my opinion, that the solution is combination of non-thermal and advanced thermal technologies. So to use the advantage of each and every technology, of course, without usage of chemicals. So, but in example of balanced usage of pH, because also we know that, in example, by lowering pH, we can also impair their ability to survive.
Thank you, Annette. Maybe one or two more questions. There's also another one that just came up in the chat. Yeah, from Eskindia sorry for that, Endalu Tadesse. Thank you for your presentation. what is your opinion on HPP regarding the color changes in muscle-based food products? What is the possible solution to reduce these changes while also increasing microbial reduction?
Yeah, so this is, in my opinion, as I know for HPP, this is the common situation that is happening because of different reactions that is happening while application of HPP. So what is really important, so we have increased like 600 megapascals while using HPP. And this is something that is happening, but it's some sensorial deviation for the muscle, so the meat products. And in my opinion, definitely the idea is to use optimization of HPP processing. So we need to make sure the pressures that we are using, the treatment time that we are using are optimal. Also, we need to check the pH, what is happening with the pH, what is happening also with the actin and myosin in these muscles. So in these products, of course. So we need to make sure what is actually happening with the hem inside of the product. But also we know that this is chemical reaction, and we need to optimize the process. This is the only way. So we need to play with the HPP, with the optimization of the parameters like pressure, treatment time, pH, salt, in example, salt percentage. So to make sure the optimal that we have optimal conditions and to avoid this deviation in colors. So what is also the possible solution to reduce the changes while also increasing microbial reduction? In my opinion, the same. So by lowering the pH, by increasing the percentage of salt. Of course, this is the common way of destroying microorganisms. But anyhow, we need to optimize the parameters of HPP, like processing time and pressure, and also the, in example, in what kind of plastic material are those muscle-based food products packaged. So what is the size? What is the compression? So I think that, in my opinion, we need to play to optimize the processing parameters.
Thank you, Annette. Maybe a short comment also from my side. Please, whenever you are looking into new technologies, and now I'm addressing the audience here, Annette, you of course know all of this, yeah? Please always have a look into the mechanisms. What are the mechanisms of actions, and what is this technology or the inactivation that is caused by a technology based on? For high pressure, for example, protein and enzyme denaturation are a big part of the inactivation. So obviously also food constituents that have a lot of protein can be affected. And in the worst case, maybe this is then not the right mechanism for the quality you want to achieve, and maybe also different technology would be more suitable then. Yeah, please, this is important to keep in mind. Okay, good. Then do we have one last question maybe? Yes, Arthur?
I do have a small question to connect with the previous presentation from the other Annette. So Annette, question for you. We talked about the previous presentation about cucumber and cantaloupe being contaminated on the surface. So people were asking about what subject could they do. So what would you recommend as a non-thermal treatment to treat cantaloupe or cucumber?
In my opinion, and from the experience, called plasma. So as a surface treatment. So definitely it can be one solution to try. And yeah, definitely to try this surface treatment. Anyhow, of course, we can try to test others.
Thank you.
Thank you.
Short one.
Good. Then thank you, Annette, very much for your insights into the non-thermal technologies. And we will now move on to another strategy on that, where we will hear more about from Uli von R from Agroscope in Switzerland. The only miracle for me, Uli, is that I have two different titles of your presentation, and I don't know which is the right one. So maybe, please, you can introduce your title of the presentation yourself, so I don't make any mistakes here.
Yeah, let me quickly share my screen. Hope you see it. Thanks for inviting me to give the talk. I mean, for me, we look a bit on the raw material for plant-based alternatives, and I give you a bit an insight on the challenges in this raw material, not inferiority. I will present some data which we have, which case we actually develop a product which we developed at Agroscope. Now, sorry. Yeah. So I will just quickly give a short introduction to Agroscope, and afterwards I will present some old acquaintances and the new guys, because I mean most of the bacteria we work with are already known in other matrices, but they may appear in the new material and other guys. And then we will look a bit on Bacillus cereus, which was also recently in the media based on a dairy case from a big company, and then I look a bit into specific challenges which come, and we'll finish with a short introduction of the horizon action for with the community of practice. I take the note to introduce Agroscope Day Research Station a bit better, because we have this year the 125-year birthday. So it exists, and actually on the top left you see the original building where our site was. It does not look anymore like this nowadays. And after 125 years, we do not only transfer from dairy research also to plant-based alternative research, we also transfer to a new building, which you see on the top right. The model, we also will have the ability to have a BSL-free pilot plant there, so we will be able actually to do pilot challenge tests in real food matrices. So for example, we can produce cheese with Listeria monocytogenes, but we cannot do it next year. It takes about 2 or 3 years to validate the operation of this pilot plant in BSL-free level. Well, for the raw material we usually use for plant-based alternatives that are often leguminous, and they are growing close to the soil, which was also presented with Annette. And there we have quite a diverse microbiota and also many spore-forming bacteria. Often those those materials are processed for the products. Nevertheless, like this, you actually increase another way to introduce pathogens to the materials, be it to recontamination, or in case of spores, they can actually really well survive this processing. What we also have, and that's a bit another topic, is the call this analytical gaps, because most of our methods are related to dairy or meat or other matrices, and then you may have false negative results or another growth behavior. And in the case, for example, of Bacillus cereus, it's still a bit difficult to identify it with cultural method. And then also for the inactivation of those pathogens in plant-based matrices or alternatives, for example, any D value or inactivation parameter so far we have for the pathogens is mostly done with classical food like dairy or meat, and we do not have any idea what happens, for example, in plant-based alternatives. And it was also mentioned in the first presentation, innovation has a high pace and is often a bit ahead in the food, with the food safety, and food safety follows behind the innovation. And I think there it's really important to perform the innovation in parallel with the food safety assessment. And I think that I go a bit ahead of my presentation. Exactly in this frame, also the community of practice aims, which was developed by Catalyze. Some hazards which were in plant-based foods, and here I really looked already on the finished processed food and not on the raw materials. So Listeria is also a common contaminant in plant-based alternatives. There was a huge outbreak about 2 years ago in Europe, mainly in France. Also in France, there were last year, I think, about 30 cases of Listeria monocytogenes cases in plant-based producing companies, which actually made it like this, that ANSES put it as third highest priority this year to look at the Listeria monocytogenes in plant-based alternatives. Also, there are studies showing that Listeria species actually grow better in plant-based milk alternatives compared to bovine milks. The same applies for Salmonella. They grow both better there, although nowadays, so far, I did not find yet any study outbreak on plant-based alternatives that were more related really to fresh produce or the products which were presented already in the first presentation. And Enterobacteriaceae, for the overall, they can serve as hygiene indicates for plant-based alternatives, but they can also, plant-based alternatives can also be reservoirs for multi-drug resistance Enterobacteriaceae or opportunistic pathogens. This was already a bit mentioned. However, for us, what we saw also in the study, one big issue for these plant-based alternatives are spore-forming bacteria. This was also already discovered in 3 years ago in a study where they looked at several raw materials for plant-based alternatives, and they found mainly spore-forming bacteria in different plant-based alternatives. Mostly that were Bacillus species, but they even also found Clostridium botulinum in those spore-forming in those raw materials for plant-based alternatives. And here it comes a bit to this so-called pasteurization paradox, because if you have spores in your product and you pasteurize this raw material, you activate the spores. You actually increase the vegetable load of your bacteria in your product. And this can, there you need to take really care of, because if you do not look for the spore-forming bacteria, this actually can lead to future problems. So I will talk about the case we had with Bacillus cereus in one of our products. Quick introduction to Bacillus cereus. It's a common bacteria. One thing is it's Bacillus identification is pretty difficult. Often you need to really look on the genome level to find the toxin production. Bacillus cereus produces 2 toxins, which are mainly known, and one of these toxins is actually very heat and pH stable. So this means also here, if you only use heat inactivation, you still have the toxin present, and if you have already a high load of Bacillus cereus, you may still have a food poisoning food, which only leads to vomiting and no death cases, but I think vomiting is also not so agreeable. One issue with Bacillus cereus is it produces very heat-resistant spores and can also form biofilms, which also makes the sanitation of Bacillus cereus in an environment or also in food very difficult. Now, my story, I begin a bit from the end. Our goal was to develop a plant-based alternative to raclette from sunflower oil press cake. The Swiss people know a bit. It was a bit in the media in Switzerland because Raclette Valley did not like it so much. Sunflower press cake is usually a side stream for sunflower oil production. It is also oil press cake is also usually also used in feed. And I also know, for example, in feed, there are sometimes also cases with salmonella, that you also find salmonella in oil press cake. In our cases, what we did, we checked the raw material, and when we did check the raw material of this sunflower oil press cake, we actually did not find so many Bacillus cereus vegetable bacteria, so it was really below the detection limit. But after heat treatment at 80 degrees, which was also related to the process, we suddenly had an increase about 2 logs of the Bacillus cereus vegetable cell count, which is really related to the activation of the spores which were present in the raw material. The numbers you see here related to considered as safe less than 1,000. This is a bit less recommendations. It's not a regulation, and it's based on dairy and other products, not really related to the plant-based alternatives. Toxin formation will appear if you have an cell count about 10 to the 5 per gram, then you risk to have a toxin production in your product. And that what we saw is when we incubated our material at 25 degrees for 22 hours, we had an increase of Bacillus cereus count up to 10 to the 8. So the Bacillus grew very well, and that was, of course, not a safe food anymore. Even during storage, we still had a high, after 56 days at 4 degrees, we still had a high count of Bacillus cereus, even though it was reduced. And most likely the bacteria which were dying were then present in spores. So they just went to spores back to survive long. Besides heat treatment, because we have a huge knowledge on fermentation and so on, we tried also to ferment the product. So we added the starter culture, which grows on plant-based, but we did not modify the raw material, did not add any sugar, and did not check. And we saw that the starter culture grew, but the pH was only reduced to pH 5.7. This helped to reduce the number of Bacillus cereus during the fermentation, but it was not able to really stop the proliferation. Only for the storage, this fermentation with the starter culture helped. However, the reason why the starter culture did not grow, it had not the correct carbon source in the raw material. So when we added glucose, so that the starter culture could grow better, the starter culture grew and the pH dropped then down to 4.9. And as you can see, we reduced actually the growth of Bacillus cereus more or less to 0. So we only kept the initial cell count of Bacillus cereus. And after storage, we even were able to completely inactivate Bacillus cereus. So for that, what we then saw in this process is that you can not only relate the heat treatment of this oil press cake and fermentation. For us, it's really important that if you could make the control of such a product, you not only need to check after heat treatment, but also after fermentation, also to make sure that your fermentation process runs well. Of course, the other parameter you can measure there is also the pH, but still you need to validate that you have reduced your Bacillus cereus contamination to an acceptable level. What we did not check in this study is the presence of the toxin. So here in this case, as we see, for example, when you did not add any any starter culture, we exceeded the level of Bacillus cereus to reduce the risk of toxin production. That we do not know. If you want to know more about what we did in this study and further data, we published it this year and it's now available to read. For the challenges which are now coming for us for the plant-based food safety, as already mentioned in the introduction, we need actually also to evolve our analytical method to the new methods, also to faster detection methods and also isolation. So if you cannot isolate your pathogens out of the matrix and they protect themselves there, you will not find them. Or in such case, also for the product like this, we need to broaden actually also the toxin broaden our toxin considerations, not only for the Bacillus cereus toxins, but also for the aflatoxins, which can be present. There was actually a recent study in meat-based alternatives where they found quite a high amount of aflatoxins. And this also goes related to inactivation technologies. The inactivation technology really needs to be adapted on the matrix. Depends also a bit on the matrix here, also in the oil press cake. One thing with the matrix, of course, is the presence still of oil, which can also be a protective matrix against some inactivation steps. And for the whole process, the HACCP concepts need actually to be adapted on the process. And what I also want to mention here, that because that was stumbled up also a bit in the literature, it does not help, for example, if we play around with safety, comparing dairy and plant-based alternatives based on the safety, because both have safety issues. It's a bit different point of view. And in terms of the development of innovative product, I think it's important that people developing new processes or new products actually start thinking about food safety already at a very early stage, and not only first at the latest. With this, I actually want to thank the people who did the work together with me, InnoSwiss, who founded the project, and also our industrial partners. And as mentioned here on the last slide, I also want to present the community of practice. This was developed by Catalyze. The idea is here really to bring together scientists, regulatory authorities, startups, and industries, so that they can help putting together food safety along the whole food processing chain, starting from the field till to the consumer. And with that, I thank you for your attention. And yeah, I'm open for questions.
Thank you very much, Uli, for the very interesting insights. Let me look into the Q&A. No questions yet. So please, the offer to the audience once again, if you want to know something on this topic, if you have detailed questions to Uli, please feel free to put it in the chat or the Q&A. In the meantime, maybe a short question from my side on the Be Serious study you showed earlier. You showed the protective culture or the starter culture you added, and then the reducing effect on the Be Serious growth. Is this just a pH effect, or is it really, or could you just add some acid and have the same effect, or is this really a protective culture effect that needs the microbial growth for it to be effective?
We did not test only with acid. The pH, they showed that the pH has an effect, certain effect, but it showed also concurrence for nutrients in place. In this case, of course, not for the carbon source, because in the original material we did not have the correct carbon source for our strain, but as you see, we were still able to actually reduce the growth of the Bacillus cereus, but not as high as if we were really able to degrade the pH. So I think it's a combined effect. I mean, this is also with any starter culture, you have the main effect is of course pH, and this you can achieve also with the addition of acid. But you have other factors. I mean, it's also known for lactic acid bacteria that they produce hydrogen peroxide or other small bacterial compounds. In this case, I don't think we have a bacteriocin producing starter culture, but could also be the case. And the starter culture, of course, modifies also your product. I mean, you have some acidity, hopefully always in a good way. If you do not want to modify your product, you can add protective cultures, which are actually only there to inhibit bacteria, pathogens or spoilage bacteria, but should not modify your product, at least not as you saw it. And I mean, the point is, if you use fermentation, and I think also any other inactivation technology, it was also mentioned for me, it's one piece of the hurdle concept. You should, in any case, hurdle concept is very important in any product. You will never be able to use only one technology to reduce the bacterial contamination completely. So, you read the question, yeah, pH has an effect, or acid alone would reduce the bacillus a bit, but here it's also a combination with space and a nutrient competition.
Thank you very much. In the meantime, one question in the chat popped up, and I would say that this is then also the last question, looking at the time. But all the speakers will be available also afterwards. We will share the contact details also. So the question from Elham Karimi in the Q&A section is, thank you for your presentation. I was wondering whether you think Be Serious could become only involved in fermented foods if the fermentation process does not proceed properly. Could unsuccessful fermentation conditions promote its growth?
Yeah. I mean,
short answer.
Yeah, I mean, the point is, if you have not a successful fermentation, it can grow. It can also outcompete your added starter culture, depending if the bacillus is more favorable. And yeah, in the end, if you have the wrong starter culture, wrong condition, you actually even can promote the growth of bacillus. So that's also why I mentioned it is really important, even if you have any heat treatment, and so you need to keep monitoring your product.
Thank you very much. And thanks again for your nice presentation and the interesting insights. Then we come now to our last presenter for today, who is Georg Kaltz from Austria, from the Bühler part that is in Austria. But Georg, I'm sure you may introduce your company a little bit yourself. And without further ado, you can already share your screen if you want to, and then all the attention is yours.
Okay. Last but not least, I hope sharing works well for you.
Perfect.
Okay. Thanks for the opportunity to giving this speech here. I would like to highlight a bit the impact of hygienic engineering and design on sustainability in food production, which sounds a bit like a niche theme. And the reason why we put it here on the agenda is that actually both Felix and I are members in EFFoST of the Working Group on Sustainability, which was quite recently founded to address the topic of sustainability also from the perspective of hygienic engineering and design. My background is with Bühler Group, which is a big global provider of equipment and technology for food production and also non-food production. And I would like to take the chance here to give you a bit of a bird's view insight of the perspective on sustainability from an equipment provider or technology provider as a company, but also from this aspect of hygienic engineering and design, which is an must-have in food production, and can really have an influence on how, let's say, sustainability footprint of food production is evolving in both directions. I will start with a short introduction of Bühler. So I'm presenting two organizations. Now, first I'm presenting a bit of Bühler, then I'm presenting a bit of EFFoST, and then have a jump into the topic of sustainability and hygienic engineering and design. Bühler Group is actually it's one of those hidden champions you have around the world, and we have roughly 2 billion people every day in contact with food that was somehow produced with our equipment, and about 1 billion of the world population are traveling in vehicles that are somehow related to Bühler equipment. So we have quite a scope. And Bühler is actually a Swiss-based, still family-owned company with roughly 13,000 employees worldwide, and has a strong foothold in many different fields. You see here, this is both the food and the non-food part. For this here, we will focus on the food industries where Bühler has a strong foothold. This is actually everything related to milling, which is actually the technological origin of the Bühler Group back in the 19th century. We spoke about plant-based alternatives. This is also a field, a strong growing field where Bühler is very active as an equipment supplier. As a bit of a derivative from milling, there's also malting. And the second big column Bühler is resting on is actually, it's a Swiss company, so we're speaking chocolate here, yeah. It's cocoa, it's chocolate, it's production of raw chocolate, it's and also producing of chocolate final products. And finally, the part where I'm coming from in Austria, that's the wafer technology. So if you have something that looks like this and it's baked and it has it's filled with some usually sweet but also savory creams, that's a wafer, and this is the part where Bühler is holds about 50% of the world market in the equipment and the technology side. So we are equipment supplier. So sustainability has two aspects for us. It's what we are doing and what will be done with the equipment we provide to our customers. And the big multiplier from the equipment producer's point of view is always the efficiency of the equipment that we provide to our customers, because there's the really big multiplication. And therefore, we have focused already some years ago on two targets. The one is for us. We try to reduce our greenhouse gases that we generate in producing equipment and solutions by 60% by 2030, which is quite a lot for a manufacturing company, to be honest. But we are on track here. And concerning the focus on our customers is we aim to have solutions available for our customers that they can reduce over the whole value chain of a product. 50% of energy waste and water in production of these products. It's our 50-50-50 goals. Yeah? And we managed to achieve this last year in all of our key value chains that we are providing with solutions. Some examples how this can be done. There is, for obvious reasons, a strong focus on energy intensive processes, whereby new technologies or better new engineering solutions for existing technologies, there's a possibility to reduce the energy needed to carry out the process step. Like we have here on the left, cocoa roasting processes. By improving the design and also using alternative heating technologies, it's possible to reduce the energy needs for roasting by up to 50%. And also in malting, where there are, for obvious reasons, there are thermal processes involved. This is possible to address here the thermal energy, the energy that goes into heat in the end. Plant-based meat is a bit of another topic. What we have to look here is that the footprint of plant-based foods is by far lower than the meat-based foods that these alternatives try to substitute. And when calculating the footprint of such value chains, like we have here, typically a burger that's with a plant-based patty, compared with the same burger that would have been a beef patty, the emissions that are generated to get this burger on the table in a restaurant is reduced by about 90% when going plant-based. And concerning this glass coating, that's actually from the technological point super interesting, but it's sadly non-food, so I will skip over this now. Second organization, EHEDG, who we are. I already told you that Felix and I are active there. That's the European Hygienic Engineering and Design Group. And it was founded in 1989 as a non-profit organization to get, actually by 5 companies, to get together food producers, equipment producers, academia, to get a common understanding on how hygienic engineering and design should work, to collect best practices and to generate, let's say, an established state-of-the-art for hygienic production, equipment for a hygienic production of food. Since then, it grew a bit. Status today is at more than 750 members. So it's a bit of growth from the 5 founding companies. And it's also not very European anymore, because actually there are members on all continents today. The headquarters of EHEDG today is in Amsterdam, but there are more than 40 regional sections around the globe where the companies are actually organized on a local level. And I told you before that the focus is on generating a state and consolidating state-of-the-art in hygienic design. This is done mainly by writing guidelines. This was more or less the first intention of EHGDG, and it's still doing it. Today, there are more than 50 guidelines in 10 focus areas that are related to hygienic design and engineering. And these guidelines are compiled by expert volunteers. Yeah? So this is a non-profit organization where interested companies and interested individuals or interested universities come together to define together what they see as the state of the art in certain fields. EHGG is also offering trainings on these topics that are covered by the guidelines, and these are typically courses that are given by EHGG certified trainers. And finally, there are certification schemes for different equipment types, and there are currently 8 authorized testing laboratories to certify such equipments that they are designed according to EHGDG guidelines. So they are seen as hygienically properly designed according to the current state of the art, and that they are properly cleanable. So what we did as EHGDG was we wrote a white paper on sustainability. What we wanted to know was, because everybody is speaking on sustainability actually is, as actually Felix said in the introduction, it is a widely used and often misused term. And we wanted to highlight here the connection between what EHGDG is defining in its guidelines and the impact this has on sustainability in food production. And we had some key positions in the beginning that we took as granted. Yeah? Point one is without hygienic engineering and design, there is no real way to have proper food safety, food quality, and so on. Yeah? So this is the core topic of EHGDG is critical for all this food production. And point two is sustainability is of growing importance for the food production, as it is for other industries. And we wanted to find the link between these two concepts. However, the core rule was that food safety shouldn't be compromised in any way to improve on sustainability. So this was not an option. Based on that, we built a concept of identifying 4 main elements we wanted to analyze concerning their influence on sustainability in food production. And this was mainly the energy that's needed for food for operation, the water that is used for operation of a food production, the chemicals that are used usually not for producing the food, but keeping food safety up by cleaning, and finally the product waste that occurs in a operation of a food producing site, even if nothing goes wrong. So it's just about product change, it's about losses in cleaning, but also we spoke a lot about inactivation today, about when there are issues in shelf life, when there are issues with incidents and recalls that lead to a waste of food that has already been produced but cannot be used because of microbiological issues. What we found quite soon was that they are very, very difficult to rank, because depending on the product, depending on the type of food and also the process step, there is a big variation in the importance of energy, of water, of chemicals. So What's not possible is to say we speak about hygienic design, we speak about food safety, we speak about sustainability. Just look at the energy, and because there are process steps where the main focus won't be energy, there won't be something else. And finally, we spoke also about the literature on sustainability and food safety, and there are not many publications yet that focus specifically on hygienic design and sustainability. So we did our best with what was available to get to a conclusive picture and to give guidance how to proceed on that. And I would now like to give you a short overview on these four elements and the four main and the main links we found there to the topic of sustainability, and also to give you a short outlook on how EHEDG is proceeding from there. Let's talk about energy, hygienic design, and food safety. The big advantage of energy in food production is that it's easily to measure. So one big role energy can play in looking at sustainability in connection with hygienic design and sustainability is that it can be used to identify process steps where big gains can be achieved concerning conservation of energy and also concerning sustainability. There will always be an energy mix on each food production site. You see there on the right a nice offshore wind park, which generates renewable energy, which is great from the sustainability point of view. But if the energy used on food production is generated by an offshore wind park or a coal-fired power plant, is usually not completely under control of the food producer. Yeah? So this is to a certain degree a boundary condition where the overall footprint of food producing operation will be strongly influenced just by the footprint of the energy it can get to produce the food. And another point is that in case of a designed equipment concerning hygienic engineering and design, and also hygienic, well-designed operations, there usually will be a reduction of the energy consumption just because of the efficiency gain the operation has. The second area we looked on was water. Actually, about 70% of the fresh water is used in agriculture. So water is important for food. Food is important for water. However, without giving any numbers, we as a species don't do this at a sustainable rate. So we're using more water than we have, and we already have serious issues because of this. So from the sustainability point of view, reduction of water withdrawal is a must. Since hygienic design is about cleaning, and cleaning is usually about water, there will be definitely a huge impact of hygienic engineering and design concerning the amount of water and also about the amount of wastewater generated in the food industry. So it really makes sense to look at this. However, there will be, as I said before, the challenge that there's a certain bias between maintain food safety, reducing amounts of water, and protect the water resources. Yeah? So this is not so easy to get all this to a satisfactory level. Chemicals, we have here CAP. For obvious reasons, an optimized hygienic engineering and design is beneficial to the efficiency of a CAP operation, getting the design hygienically done will reduce the amount of CAP that's needed, which will usually reduce the amount of chemical that's needed and can reduce the amount of water that's needed. Same challenge as water. Food safety still has to be given. And finally, product waste. What you see on the right side, that's red, that are tomatoes. Behind the tomatoes, you see the greenhouse where the tomatoes were. There's estimations that about 1/3 of all food that's produced on the planet is wasted in some way. And the food we produce on the planet is responsible for about 6% of the greenhouse gas emissions we have on the planet. So for obvious reasons, reducing the food waste is a thing that will have a high impact on sustainability on a global level. So of this, of the food that's produced, about 15% is lost because of poor processing, storage, and handling, meaning there is quite a big lever in the food industry to become more sustainable by getting this quota of lost food down, as we can see in the big numbers on the greenhouse gases. And the contribution of hygienic engineering and design here is that there's a contribution to material utilization, meaning less loss in production by proper hygienic design. And also, if hygienic design is done well, this is the prerequisite for a reduced risk of contamination and recalls, which will also lead to a reduced amount of food that is lost. What's next from the EHGDG point? I told you before that we found that there's not much literature there that really is dealing with hygienic design and sustainability. There's quite a lot of literature on food industry and sustainability, but hygienic design is usually something that's not covered there in detail. So EHGDG will start a joint project with the University of Limerick to develop a framework for sustainable hygienic engineering design, and also its validation to get numbers on what impact hygienic design can have on the sustainability of a food processing unit. And I told you we wrote a white paper on sustainability as a sustainability working group. So this was the first time that sustainability really was in focus on EHGDG, and there are 50+ guidelines already there. And what will be started now is that each new guideline EHGDG is producing will have a chapter addressing the sustainability aspects of the topic of the guideline. And for the existing ones, which are quite a lot, there are update cycles in EHGDG. They aim to update it every few years, or at least to re-evaluate it every few years. And there we will now include in this re-evaluation and update process also the aspect of sustainability, so that the already existing guidelines in EHGDG will also address this. And we're looking forward to get input from this joint project to have there enough data for this updating of the guidelines. With this, I thank you for your attention. It was a pleasure speaking to you on this. And if there are questions, I'm looking forward to it. Or my contact details are available, so I'm also available offline of this webinar on that.
Yeah, thank you very much, Georg, for sharing these insights related to hygienic design and sustainability. Since we're a bit over the time already, maybe just one quick question, and then, as you said, people can get in contact with you afterwards. Yeah, maybe a short question from my side. At the moment, when we look into cleaning and disinfection, everything is very much schedule based, right? How would you imagine a future where these aspects can be sustainably integrated in a food processing factory, and how could this be achieved?
I think that one thing we will need is a stronger focus on really measuring processes, also measuring cleaning processes. Because if we stick to, if we don't get a certain, let's say, flexibility on seeing when a certain level of reduction is achieved, it's the same. It's the same when we speak about we had the non-thermal inactivation technologies, the thermal inactivation technologies. What we see here is that there is really a way to improve on that when we get live measurement of processes, maybe cleaning processes, maybe inactivation processes, and based on that, really start to control these processes concerning time, concerning parameters. Yeah. And this is to a certain degree, it's always about measuring, it's about data, it's about understanding these processes. And I think one thing we have to keep in mind in especially when we speak about water and chemicals, there is always the question on what the motivation is in optimizing a process. And when we speak about water, water is quite often not expensive. From the monetary point of view, water and saving water is an issue from the sustainability aspect, from the environmental protection aspect. So this is also a question about awareness and also about the legal framework companies are operating in. Another point that we have to keep in mind here is I told energy is easy to measure, yeah? Because in the end, everybody gets a electricity, a bill for electricity, a bill for whatever, yeah? And they're usually big numbers. And so they are monitored, they are measured, they are optimized. The bill for water is usually quite low compared to the bill for electricity. So there won't be so much activities just from the monetary point of view. There won't be so much so much data. There won't be so much insight into the processes. And I think this is the key for all also the processes on the plant-based extrusion that Uli brought, or the thermal inactivation that Annette brought. The key topic is here really measuring the processes, understanding the processes, understanding the interaction between the process and the product, and optimizing there.
Thank you very much, Georg. So we still have a lot of room for discovery, let's say, and for new developments. And I'm looking very much forward to see how this will develop in the future. Yeah, once again, thank you very much. And also thanks to everyone who is listening. Just one last announcement before we can close this webinar. You know that from the EFFoST side, we are regularly organizing webinars. We have different working groups, and the next webinar will come up in the end of September, organized by the Food and Health Working Group. And then it will be about alternative proteins and their effects on the human health. Yeah, related, for example, to the second meal effects. How does a meal you consume influence the next meal you take in? But also aspects related to different process sources, how this can affect your health and your digestion, the gut microbiome, metabolic health, and similar aspects will be covered there. If you're interested in this topic, please stay alert. Yeah, follow EFFoST through the newsletter, through LinkedIn, and then you will get updates when the registration is open. And if you want to follow closer our activities from the EFFoST Sustainability Working Group, you're also very welcome to join us either on LinkedIn. Here we have a group. That you can join on LinkedIn, and also if you have particular questions or input, maybe you have a certain topic you would like to hear more about, you can of course also suggest topics, for example, for webinars and similar things. So you can also shoot us an email at sustainablefoodsystems@effost.org. And yeah, we are looking for your input, because then of course we can provide webinars that are particularly interesting for you and your target group. And with this, I would like to thank, first of all the speakers, everyone who was involved in the organization, Arthur, Lan, and Anna from the EFFoST head office, all the speakers, all the participants. Thank you so much for staying with us longer than expected on these interesting topics. And I wish you all a nice afternoon now, and I'm looking forward to see you again at the next webinar. Thank you all once again, and goodbye.
Thank you.
Thank you.
Thank you.
Thanks.
Bye-bye.
Thank you. Goodbye.
Bye-bye.
Bye-bye.